Alright, so last time we talked about hematocrit and introduced red blood cells. So today we're going to talk about red blood cell synthesis, also called erythropoiesis, right? So this basically is the generation or the genesis of red blood cells. So this process occurs primarily in the bone marrow, where a multipotent hematopoietic stem cell or a pluripotent hematopoietic stem cell, which can virtually divide into any cell of the blood lineage, is activated. So when this multipotent hematopoietic stem cell is activated via its receptors, it matures into a myeloid progenitor cell. So this myeloid progenitor cell can be of two forms, right? It can be of the best forming unit, which is an erythroid, or the colony forming unit, which is also an erythroid, right? So this myeloid progenitor is activated by a very important hormone or a very important regulator called erythropoietin. So this erythropoietin activates the myeloid progenitor via its receptor, and it matures again into a pro-erythroblast, right? That is a precursor to or it's a immature red blood cell, a pro-erythroblast, right? So this pro-erythroblast still contains a nucleus, and it further matures into a basophilic erythroblast, which matures again into a polychromatic erythroblast, which then again matures into a orthochromatic erythroblast. When it reaches this stage, this erythrochromatic erythroblast undergoes enucleation, which is the removal of the nucleus, right? After this removal of a nucleus and the endoplasmic reticulum, we then form a structure called a reticulocyte. So this reticulocyte is the precursor to the erythrocyte. This reticulocyte has a lifespan of 1 to 2 days before it matures into the erythrocyte. So before it matures into the erythrocyte, firstly, we see that these RNA molecules are removed and flushed away, right? This reticulocyte will still contain some structures, but before it matures, these structures are removed, such as this RNA, right? After the removal of these structures, that is between 1 to 2 days, they then mature into erythrocytes or red blood cells. So these red blood cells are biconcave discs, as we said last time, which do not contain a nucleus and mitochondria. They are cells which undergo anaerobic respiration and have a lifespan of 120 days, right? And these erythrocytes, they do not have regenerative capacity, so they must be continuously broken down and made, right? They are broken down by the spleen and liver and remade again by the bone marrow. So that's all on red blood cell synthesis. In the next video, we're going to talk about red blood cell regulation by this hormone called erythropoietin. Thank you.